Okay, I think we'll start. Okay, welcome to week two of Advances in Omics. Today's session will be a little bit repetitive with respect to the first one because we'll be discussing uh, pyrosequencing again, mostly, and other small sequencing techniques. So I thought a lot of you were asking me in the last session to discuss a little bit about how to do the data analysis. So after I complete the topics for this week, I am going to briefly talk through the different kind of algorithms and techniques that are used for data analysis for genomic data, right? Okay, so we have a few things to, good amount of things to discuss today, so let's get started. Okay, yeah, as I was saying, that we'll start with the pyrosequencing module. We have gone through it a little bit in the previous session, but we'll do it again in a little bit more detail. So, yeah, the main idea of pyrosequencing was to detect addition of or incorporation of nucleotides with the help of different fluorescent enzymes, for example, luciferase. So, that was the main pyrosequencing technique. Now, if you parallelize that pyrosequencing technique through different in a single plate through different flow cells, what will happen is you'll increase the throughput of the technique, and so parallelized version of this pyrosequencing was developed by 454 Life Sciences, and after that later acquired by Roche Diagnostics. So right now, pyrosequencing is under the license of Roche Diagnostics. So the basic idea which we discussed in last session as well. So you have the DNA, you amplify the DNA inside water droplets in an oil solution, which is an emulsion PCR, with each droplet containing a single DNA template attached to a single primer. And then it forms a clonal colony according to these attached DNA templates. And what this sequencing machine does is it contains many picoliter volume wells, each containing a single bead and sequencing enzyme. So inside each well, there is the oil emulsion, inside which there is the DNA template attached to a single primer, and inside that whole well, there are the rest of the sequencing enzymes which are going to going to like so there will be polymerases, there will be other enzymes which include luciferase and other phosphate enzyme, phosphatase and kinase enzymes as well. So generally pyrosequencing will use luciferase to generate the light for the detection of these individual nucleotides. So once you add an individual nucleotide, you will get a signal, and according to that signal you can determine which nucleotide has been added to the nascent DNA, and the combined data is used to generate the readouts, which are the reads basically for the sequencing experiment, right? So generally these are like the general facts about pyrosequencing. So generally the read length base pairs are around 200 to 300 base pairs, but later on, and also like during which pyrosequencing was used, we have developed techniques to increase the baseline. So it can go up to around 1 kb, a little less than 1 kb. So Sanger can go up to around 700, 800. This can go up to around a little less than that. Uh, but this is more than what Illumina gives. Illumina will give you around 300, 350 base pairs of plane. So it is slightly more than that, but these are like older estimates, what you find in the literature. But right now it kind of sits in between Illumina and Sanger. So if you do parallel sequencing, you have around 400,000 reads of parallel sequencing, 100 MB of output per run. And around, if you want to run one human sample, it will be around 7.5 hours it takes, right? So the principle, I'll discuss the principle of the sequencing technique in the later slides as well, but little bit of overview. So first you have the nucleotide incorporation. So when the DNA is getting synthesized, a DNTP from the solution is added to the 3' end of the growing strand, and this process will release the pyrophosphate of phosphate as a byproduct. This pyrophosphate will go and catalyze ATP. So ATP sulfurase will catalyze the reaction between this pyrophosphate, and there will be adenosine phosphosulfate in the solution as well, which is the APS. So the pyrophosphate and the APS will react with each other, and this will be uh, catalyzed by the ATP sulfurase, which will produce ATP. So you have APS, or in some cases you have an ADP diphosphate enzyme. You have PPI. They react with each other and gives you the ATP. Now this ATP will go and basically convert this luciferase enzyme into luciferin and other substrates, which will give the visible light, which will give out the signal, which can be read by some signal detector, which can be then transformed into what base has been incorporated in that particular position. One thing to note is the intensity of the light is proportional to the number of nucleotides incorporated. So if you have a homopolymer regions of the genome are regions where you have the same nucleotide repeated multiple times. So generally we have short homopolymers, which are like around 4 to 6 base pairs, and we have slightly larger homopolymers greater than 10 or 11 base pairs. So a same A repeated 10 or 11 times. Now when this will be happening, it is very so how is the signal seen? So if you have A, there will be a peak. If there is T or anything else, there will be no peaks in the rest when you move to the right. And again, when there will be a peak, right? Now, what happens if there are multiple A's, one after another in a long homopolymer stretch? So the signal will go up, but the intensity of the signal will determine how many bases are there, because the signal is the same for all the nucleotide for all the same nucleotides, right? So if it's an A or a T or a G or a C, if there are repeats of the same nucleotide, the signal is the same. It's the intensity of the signal which will directly tell you how many bases are there at one repeat unit. Now the issue with this is prone to errors, not only uh, in case of pyrosequencing. If you even do long read sequencing with Oxford nanopore data, there also in real life also, when we do handle Oxford nanopore data, we see a lot of these homopolymer errors. So if there had been 7 or 8 nucleotides uh, of the same nucleotide repeated one after another, nanopore might count 7 instead of 8, 5 instead of 6, things like that. So one less or one more it can count. So if that happens, in this case also, if that is happening, what will happen is when you do the later data analysis, when you take this data and do alignment with the reference genome, and then you call variants, so spurious indels of 1 or 2 base pair length will be incorporated because the number of bases have counted was less or more, right? So depending on that, insertion or deletion will be incorporated, but that will be a spurious variant. Because that variant does not exist, you are only finding that variant because the sequencer uh, has not counted the number of repeats of the same nucleotide in a perfect manner. It has either underestimated or overestimated. So you get an indel around that region. This is an issue which is still we face with even modern third generation sequencing technologies as well. But this issue you can also see in pyrosequencing. Okay, so after you get the signal out, then there is also cleanup step where this enzyme A pyrase is introduced to degrade any unincorporated DNTPs, which will prepare the system for the next cycle. So before the next cycle with the next set of nucleotide, you have to remove any unincorporated DNTP from the solution, which is done by this enzyme A pyrase. Uh, okay. So a little bit about how the sample is prepared, and then how actually the sequencing occurs. So the first step, there will be PC. You have to amplify the DNA. So there will be the first step will be PCR with biotin labeling. So one primer will be biotin elated, resulting in PCR products where one strand will be tagged with biotin. So you have the stretch of DNA. You add a biotin uh, uh, to one of the primers. You add biotin, and then you do PCR. So along with the DNA, the biotin will also be so so once you amplify the DNA, across every DNA strand, there will be one strand containing the biotin labeling. Then you capture, the important part is, why do you have this biotin labeling, is to capture the biotinated PCR products on streptavidin coated magnetic beads, anchoring one DNA strand firmly. So basically, it's what we use in Illumina, where we use those. If you remember the video that we saw in the last session, there is always a template primer strand attached to the flow cell, where and you also attach an adapter to your basically read, and that read will come and bind to the flow cell adapter, because the adapter that is added to the read and the primer in the flow cell will be complementary, so they'll come and attach it there. Similar kind of functionality is seen here. Instead of using a DNA adapter, here they use biotin, and then you use streptavidin coated magnetic beads, which will kind of pull the biotin, which will get attached to the biotin, and you'll have uh, kind of uh, basically a DNA strand which is stable present in one place. Basically, it is keeping the DNA strand in one place. The next step, just a second. Yeah, guys, again, I'm going to request you one more time is to please join uh, within time, or else like it becomes very difficult for me to switch between the slides, and I have to admit you in. I have no other option. Like I cannot just keep it open to everybody. So I have to admit you in. So if you join at random times, it becomes difficult for me to navigate between these two things. So just a small request to all of you. Please try to join within time. Okay. The next step after you have the biotin-related DNA, which has been captured by the streptavidin BIDs, then the next step would be to denature this double-stranded DNA into single-stranded DNA, right? So what would happen is you use alkaline denaturation, so something of like sodium hydroxylase, NaOH. You use an alkali like NaOH to just denature the double-stranded DNA into single-stranded DNA. What happens is the non-biotin-related complementary strand is released and removed, while the biotin-labeled strand will remain bead-bound. So you can imagine. So you have a bead. So I think you can see it from here. So this is a double-stranded with DNA. In one strand, the biotin is attached. This is the streptavidin beads. So this biotin will come and attach to the streptavidin bead. Then you do give the NaOH. So what will happen is it will denature this double-stranded DNA, and only the strand which is attached to the streptavidin bead will remain, and the other will will be washed away. So that is generally how you take the sample and get the DNA, right? The next step would be the sequencing setup, where you will transfer this into a microtiter plate, and the bead bound to the single-stranded DNA is transferred into the wells of the sequencing reaction. Then you add the sequence enzymes. We are going to come to this in the next steps. Then you're going to add the sequencing enzymes, and then you kind of do the sequencing and capture the signal with help of some signal detector, right? So this is how it is, how the DNA is prepared for the sequencing step. Uh, yeah, I've just quickly go through the sequencing step again. We have done this previously, but again. So the preparation will be placed in the machine. The primer will be first hybridized on the template, then followed by a gradual DNA synthesis. It will add one of the four necessary nucleotide triphosphatase, and therefore, after that, like with the addition of the one DNTP, uh, it will release the pyrophosphate molecule, which we discussed, and then this will react with the luciferase enzyme, and no, sorry, pyrophosphate will kind of generate ATP. The ATP will react with the luciferase enzymes to produce luciferin, which will give you the light. And that light production will be recorded during the entire procedure by means of a specialized camera, and it will be quantified, evaluated, and presented in form of signal peaks, which will be the work of the base caller. And as I was saying, if there are two or three of the same nucleotides which follow one another in the template, several nucleotides will be installed in a single node, and the light production will be twofold or threefold as high as normal. So basically, the intensity of the peak will tell you how much or how many number of nucleotides you have, which I was telling you the issue with homopolymers. So if you have 7 or 8 nucleotides of the same nucleotide of the same base one after another, so generally if you have 8, the idea would be the the signal intensity that you have from one peak times 8 will be the signal intensity if you have 8 similar nucleotides one after another, right? So yeah, so this is a little bit error prone. And the idea is that if you have no nucleotide inserted, it will remain pitch black. And yeah, so and if you have surplus DNTPs, and if you want to complete the reaction rapidly, the DNTPs can be broken down into DNDP and phosphate by adding the epidase. The epidase, as I was saying, is adding in the last step after the round of sequencing has been done. So to remove the extra DNTPs, right? So yeah, I'm not going to go into this slide again. We have discussed this in the last session, but I'm going to discuss some other aspects of the sequencing part. Right. So uh, after yeah, now this is basically the kind of sequencing procedure are followed in these 4 steps. So once you have reached this particular step, uh, you have denatured, you have washed away the extra set of DNA strands, you are left with this particular structure. You have the streptopyridine beads, you have the biotin attached to it, and the DNA attached to that biotin. Right. So now in the next step, the polymerase will come into the picture and start the catalytic reaction using the free 3' end of the 3' hydroxyl end of the primer. It will incorporate the first nucleotide, and then it will extend the primer adding one nucleotide after another. The nucleotide analog will contain a fluorochrome and a blocker on the terminal molecule for detection and termination respectively. The terminator will be present in the 3' end and will block any further extension. And the polymerase will remain there until and unless it has reached until basically uh, the whole detection is complete. So as I was saying, before detection, you have to wash away the first, like the single-stranded DNA that you have denatured, right? This will remove any unincorporated and unused nucleotide to avoid signal disruption. And then the fluorophore is released from the nucleotide, and using a laser light, the fluorophore is emitted. There will be a high-definition camera which captures the image and is detected by the detector. And yeah, and after that, if you want to remove it, immediately after the fluorescent signal has been detected, uh, the terminator or the blockal molecule will have to be removed by washing the nucleotide. And now the 3' end is ready for another round of synthesis. So basically, the difference with Sanger here, if you remember, in Sanger, the moment you add a DDNTP, the sequencing stops. So when you're doing sequencing by synthesis, you're detecting the sequence by adding nucleotides one after another. But in Sanger, when you add that DDNTP nucleotide, the sequencing stops. Here, there is some sort of mechanism by which reversible termination can occur even after addition of the after the fluorescent nucleotide. You have gotten the signal. After that, you can keep on adding the nucleotides. It does not stop there. So that is a major difference with Sanger, and this technique is still followed in today's NGS techniques like Illumina also, where the termination does not occur immediately after the special nucleotide has been added. Even after it is added, you can just remove the extra blocker. You can just keep on adding one after another. right? So this is what is known as reversible termination. So once you add, you block it for a certain amount of time, which will be very small. Then you detect it, and then you remove the blocker. That is how generally it works. And this is the concept of the reversible termination, right? Okay. So I'll go through a quick recap of the other sequencing techniques as well. So we discussed about Maxim Gilbert. This was one of the first ones. Then we discussed about Sanger sequencing. We have not discussed about sequencing by hybridization, right? So in this case, oligonucleotide arrays detect matches to known sequence, which is fast in principle but poor in mismatch, single mismatch discrimination. So basically, this is not directly sequencing, this is only detecting mismatches. So I don't know if anybody of you have heard about arrays, microarrays, sequencing arrays. So I just quickly, I have from an older course. This is the, yeah, just quickly go here. So the DNA microarrays will look something like this. So it's a plate, and inside plate there are wells, and inside each well there is a DNA probe, which represents a single gene, or it can represent uh, some other regions of the genome as well, but it will be very targeted, right? So what will happen is there is if there is a probe inside that well, and if your read goes and binds to that probe, if there is a perfect binding of all the nucleotides getting hybridized to form the double-stranded DNA, it will emit a signal which will be captured by a kind of a high definition camera. And again, similarly, you can tell if there is a mismatch. So generally, this microarray is used to detect single nucleotide polymorphism. So basically, uh, places in the DNA where it differs by a single or two, three bases at one stretch, right? So if there is a mismatch, the signal that will come out from the microarray will be very distinct, and you can detect that there is a signal there. There's a mismatch there. So generally, this is how microarray technologies work. So this is not NGS sequencing. You are not sequencing each and every position of the genome. What you're doing is, you know that this position of the genome, you know the sequence of that. You put the sequence of that with some mutations in the genome. You can detect those mutations, right? You cannot detect all the bases, but if there are any mismatches in the base, you can detect that. So that is the general idea of DNA microarrays. Microarrays are still used to date for genome-wide association study, where you just want to see across thousands and thousands of samples what are the mutations, what are the single point mutations, right? So in those cases, you use microarray technologies. It is also used for mRNA sequencing. So that is also again not sequencing. You use mRNA to say you when we quantify gene expression, what when we say that gene expression is higher or lower, generally, we mean the gene expression by the concentration of the mRNA produced by that same gene, right? Now, if you have a probe inside a microarray which can detect which will give you a signal which will be proportional to the number of mRNAs for that particular gene. So if you have detected 5 mRNAs, you know that is a baseline expression. If you have detected 20 mRNAs, that means a gene is getting overexpressed, right? So that is also used. Those kind of uh, analysis is also done using microarray technology. So this is quite important. The difference with sequencing here is basically you do not generate the whole sequence. You only generate at which positions the sequences are different, or if the intensity or basically the number of reads of a single stretch of DNA or mRNA is getting overexpressed or underexpressed. Those things you can detect very well with uh, microarray technologies, right? Okay. Uh, Now coming back, then again we have sequencing by synthesis, where you add basically nucleotides with the help of polymerase, and you give out some signal, be it uh, fluorescent signal, be it some radioactive signal, whatever. You use that signal to tell what base has been incorporated at that position. So generally, these are the basic ideas on which most of the sequencing techniques are based. So yeah. the principle of the pyrosequencing technique, as I was saying, that you have at step one is basically you add only one DNTP at a time in a cyclical or programmed manner. In the next step, the polymerase will extend the strand if the base pairs correctly, and it will release a pyrophosphate. There will be ATP sulfurase in the solution, and this pyrophosphate will bind to this APS enzyme, which will convert it into ATP. Now, this ATP will react with the luciferase enzyme in the solution, and it will produce luciferin, which will be emitting visible light, which will be captured by that high definition camera, which you will have. And in the next step, you have to wash away the leftover nucleotides using apyrase. Okay, so these are the four basic coupled enzymatic reactions. I think we have discussed this enough, but like if you just want to take a look, yeah, you have DNA, you use DNTP, you get this pyrophosphate out. This pyrophosphate will bind with APS to give ATP. Now this ATP will bind with luciferin to give luciferyl, and then luciferyl AMP will be converted to oxyluciferin, and light will be emitted by luciferase. These are the basic four coupled enzymatic reactions for pyrosequencing. A little bit more information on the enzymes particularly used and from where these enzymes come. So the polymerase, it's just a special type of polymerase known as Linau polymerase. It comes from the E. coli DNA polymerase 1 fragment. ATP sulfurase is basically coming from yeast. The luciferase is a very specific enzyme found in fireflies, which emit light. So the enzyme is particularly found there in fireflies. And apyrase is generally coming from a potato tuber and other organisms. So yeah, so all of these are not human enzymes, just to keep in mind. Okay. So yeah, I've given some extra information about the enzymes itself. These are like not very relevant to you all, but this might, like, if anybody has interest, who might want to go through all this. I'm not going to go into details of these, but yeah, these enzymes are quite important, and yeah, these are non-human uh, enzymes. The next question would be, why is there a limit on the read length? For Sanger, we understood because you're doing sequencing by synthesis, and you are kind of uh, generating, you're knowing what the nucleotide is at that position based on the radioactivity signal or the fluorescent signal after that nucleotide has been added, right? Here, there the issue was, if you go beyond a certain limit, say for example 700, 750 base pair, the resolution of differentiation between 751 and 750 will be very low because you're going to do it in a gel electrophoresis. Remember, so you have the DNA fragments, you run it through a gel electrophoresis, and based on the length, it will stack up, right? And based on the stacking up, you can uh, get the full sequence. But the issue is that if you increase the read length, it is very hard to discriminate between two reads of very close lengths, right? Uh, Here, the issue is slightly different. What it happens is, so the background sequence, which will be the primer miscellaneous, produces spurious signals that appear early in the run, especially on templates longer than 600 base pairs. So here the error issue comes out. Here it's not the resolution issue. Here it's basically spurious signals can be generated or around 600 base pairs, right? So there are two more types of errors that gets incorporated when the uh, read length exceeds a uh, certain limit. So first is the plus frame shift, and the second is minus frame shift. So in plus frame shift, what will happen is incomplete apyrase degradation will leave residual nucleotide, which will let a fraction of the nucleotide templates race ahead of its majority. So what will happen is it will not destroy all the residual nucleotides will keep some of the residual nucleotides, which will have some competitive advantage because those are already attached to biotin related things and so on and so forth. So basically, it will introduce errors. So between each nucleotide dispensation cycle, this APIDase will be used to degrade unincorporated DNTPs and residual ATP before the next base flows in. If that degradation is incomplete, a small leftover pool of the previous nucleotide persists into the next cycle, and uh, some fraction of the million of clonal template copies on a bead will incorporate this leftover base in addition to the new one being dispensed. So particular strands get one base ahead of the rest. So basically, it will shift the frame by one base pair, right? So it's a frame shift mutation that will be seen, but it's actually not a frame shift mutation. It's a sequencing error. Second is a similar, the outcome is similar. There also frame shift occurs. Here, this one I have already discussed in terms of homopolymer stretches. The same nucleotide repeating five, six, 7 times. But again, since the intensity will be proportional to the number of bases there, that it is sometimes erroneous. So it cannot fully distinguish between, say, a 7 nucleotide bases and a repeat stretch versus a six nucleotide repeat stretch. It will not be able to distinguish it very well. This, as I am saying, is also an issue with like current long read callers as well, specifically Oxford Nanopore Technologies. So there, here, since you're using fluorescent signals to tell what is the number of bases or what base is there, right? In ONT, anybody wants to say anything? Yeah, Apurva, the slides will be shared. If you have any questions, do like raise your hand or like just unmute yourself and ask. I might not be able to check the in-call messages all the time. So yeah, let me know if you have any questions. Okay. Yeah, as I was saying, here you're measuring the fluorescence signal, right? In Oxford Nanopore, you're going to measure the current flow, how much of the current flow is getting blocked or how much of the current is actually flowing. Based on that, you determine what base is there. And if you have a similar kind of a situation, multiple of the same bases repeated multiple times, there also, since you are basing your signal, your base calling the number of bases in that location based on the intensity of the signal, it can be erroneous at times. The intensity might not the intensity might look like there is 6 bases, but actually there are 7. Things like that. So there also this frame shift will occur where instead of counting n number of nucleotides, you're counting either n minus 1 or n plus 1 or n plus 2 or n minus 2. So a shifting of the frame is occurring. So that is also limiting the lead length. If you increase it beyond a certain point, which is around like 500, 600 base pairs, these errors keep on compounding and the data that you are generating is not of good quality. Okay, so one way to protect against this is using SSB, so single strand bases. Uh, uh, so basically, there are these SSB enzymes which bind to single-stranded DNA and kind of stabilize them, and it protects the single-stranded template from going into secondary structure or and non-primer specific annealing. So if you do that, it increases the base length. It can go up to 800, 900 base pair, but uh, here also it will not be like foolproof. There can be errors. It's just that you can get a better throughput and a better read length if you use SSB. This is kind of a, I don't think this plot is based on real data, but this is generally how uh, the the throughput will look, the read length will look with with and without SSBP. So single strand base stabilizing proteins. These the same SSBPs are used in DNA replication in the actual inside the cell. So when you have a double-stranded DNA, you break it down into single strand, and then you have to stabilize this single strand before you use DNA polymerase to do the replication. So there also SSBP proteins are used. Okay. So yeah, that was mostly all the discussion that I had with respect to pyrosequencing. The next type of sequencing that we are going to talk about is ion torrent sequencing, right? So here, this idea is very kind of similar to newer long read technologies like ONT. It's a similar kind of idea in terms of detecting the base. So the idea is when a complementary nucleotide is incorporated into a DNA, a phosphodiester bond is formed, releasing a hydrogen ion, which will change the pH of the medium. The ion sensor converts this chemical information, how much of pH has changed, into digital electronic information, which will tell you what base has been incorporated. So in as in other kinds of NGS, the input DNA is or RNA is fragmented around. This is of shorter length, around 200 base pairs. Again, adapters are added, and one molecule is placed on a bead. The molecules are amplified on the bead by emulsion PCR, similar to pyrosequencing, and each bead is placed into a well of a single well of a slide. So across all the technologies, if you notice, the main differences you need to remember are how you are generating the data, how you are telling that there is this base at this position. From Sanger, you did it with respect to termination of sequencing, and then you run it on a gel electrophoresis. In pyrosequencing, you used uh, fluorescent labeled enzymes, and based on the signal of the fluorescence, you determine what base pair is there. In ion torrent sequencing, you determine what base pair is there with respect to the change in the pH of the medium. So each nucleotide incorporation will tell you distinctly what change in pH has occurred in the solution, and if you can detect that, you can know what nucleotide has been added. Okay. So yeah, this is the schematic of how it works. So yeah, you have this, you add basically one DNTP at a time. Yeah, this is important because the throughput is an issue here, because if you um, if you use multiple wells, then only this. So basically, if you want to have high throughput of this particular technology, what you need to do is you need to have singular wells in a plate. Inside each well, you're doing this technique. You're adding one molecule and the pH is changing. And inside each of these well, you need to have pH sensors to tell you what pH has changed and what base has been incorporated. So, which is very difficult to design. So, in a lot of the cases, the throughput of ion torrent is kind of a little bit on the lower side compared to the other ones. Okay, so in like 454, the slide is flooded with single species of DNTP along with buffers and polymerase 1 NTP at a time. The pH is detected in each of these wells as each hydrogen ion is released and will decrease the pH. The change in pH allows us to determine if the base and how many thereof has been added to the sequence read. So yeah, generally that is the idea. And based on the pH change, you'll have these peaks which will tell you what exact base has been added. Right? Yeah. So this is I have not sorry, I have not uh, put the heading here. This is basically the solid technique, so LID, which is basically sequencing by ligation. I'm not going to very details of this, but yeah, a general overview of how the workflow works. Right, so this is basically sequencing by ligation. It's not synthesis. So it will use fluorescently labeled probes to detect dinucleotide pairs, not individual bases. It will detect the ligation. It will not detect the addition of each of these bases. So generally, how it is done is basically you do the to fragment the DNA and prepare and amplify the DNA, which is kind of common across all the technologies. Then what you do is you attach these DNA fragments to beads and you amplify using emulsion PCRs on the beads. Probe then you do is probe hybridization and ligation. So basically it will contain known bases and a fluorescent label. So it'll be a stretch of DNA probe which will go and ligate to the single-stranded DNA, and there will be a fluorescent label attached to it, which will emit the signal once it is attached. And DNA ligase will join the matching probe to the template. Instead of DNA polymerase here, in which we were using in Sanger, uh, in Roche, in other techniques in Illumina also. Here, DNA ligase will be used because you're just attaching two probes, right? You're not you're synthesizing anything, right? You're not synthesizing anything. So you need DNA ligase instead of polymerase. And then once the ligation is done, the signal will be detected. And then again, you do this. You cleave it off, and then you again start a new cycle. You detect this. Right. So this is basically the overview of solid sequencing technology. This has been, I think, quite well discussed in the class as well. Okay. A final type of sequencing that I'll be talking about is polymerase sequencing. Yeah. Here, the principle is slightly different again. So it will polymerase sequencing works on the principle of amplifying and analyzing DNA within a solid matrix or on beads to allow accurate and simultaneous sequencing of millions of fragments. So what it'll do is it'll sequence the DNA within a solid matrix or the DNA that is attached to a particular bead. So if you can hold these stretches of DNA in place across different wells or across different lanes of your flow cell, then you can create these tiny clusters of identical DNA molecule, which are called as polonies. And these are immobilized within a matrix and serve as template for sequencing. Each colony will represent a single DNA template, ensuring the amplified sequences remain distinct. This prevents from mixing or contamination between DNA fragments. So basically, each colony is a cluster of uh, similar sequenced DNA sequences of very similar sequence. Yeah, so each polymer will represent a single DNA template, ensuring the amplified sequences remain distinct. This will prevent from contamination and mixing with other DNA fragments. And then sequencing will be performed on these polymers, these clusters of similar sequence DNA strands, using fluorescently labeled nucleotides to identify each base. So this process begins with constructing a paired tag library from genomic DNA, followed by clonal amplification on microbeads. This kind of steps, preparing library, amplification using beads, these are very common across technologies now. You can see that. So these techniques you have to follow in, in, in these techniques. After that, how you sequence the actual read is kind of different between different technologies, but the basic idea behind it is mostly similar. The amplification produces discrete polymerase colonies that serve as the basis for sequencing. The amplified beads are then enriched. The enriched beads are embedded in polyacrylamide matrix, creating 2D arrays that allow simultaneous sequencing of millions of DNA templates. So yeah, the quick takeaway from this technique is the use of these clusters of DNA together, which is a polyny. So that is the main USP of this technique, right? Other than that, you again put it on a bead, you put it in a particular well, then you use polymerase uh, to kind of sequence it and use fluorescence to detect what sequence is going in. So those are kind of similar. The one USP of this is use of these polynes, right? So yeah, that's all in terms of all the things that I wanted to discuss for this particular week. Now, what I'm going to do is before I go into the assignment questions, I'm just going to quickly give you an overview, maybe 20 minutes around I'll take, of maybe a slightly bit more of how we analyze the data that is generated through these sequencing techniques, right? Okay, this is an older slide I'm using. Sorry for that. Yeah, we're going to talk about hi-fi pack bioreads and Oxford Nanopore in later classes. I'm just going to talk about what happens after you generate the data, right? So I'm just going to start from here. Mm, Yeah. So let me talk a little bit about variant calling and alignments, right? So generally, what you do is after sequencing, what you get in terms of the data is a FASTQ file, as I showed you last week as well. Just let me quickly bring it up. Yeah, you get a file something like this. So let us assume you have done Illumina, which is the most used technique currently. So you get a FASTQ file out of it, which is basically not very complicated. It's a stretch of these reads, right? So you know these are the quality parameters below. This is the ID of the read, and this is basically what the read has in terms of the sequence. So generally, what you get is these set of reads. So these sequences, random sequences of reads. There will be multiple of these reads, and now you have to say where these reads have originated from in your own genome. Right. Doing that is quite difficult, and that process is known as alignment. So how you do that? You take a reference genome. So what is a reference genome? Reference genome is a known human genomic sequence of so human genome is around 3 billion base pairs long, right? But all of it is not resolved as we were discussing in the last session. Right now, through T2T, around most of it is resolved. Still, it is not fully resolved, but like we can assume it is fully resolved now. So basically, you have 3 billion base pairs in the DNA, and the sequence of these 3 billion base pairs has been determined through multiple years of experimentation. So the first it started with the Human Genome Project. After that, as the technologies emerged, we had newer and newer builds of these reference genome, which is going to tell you from base pair 1 to base pair 3 billion what is the sequence of nucleotides, right? So that is generally how a reference genome looks like. So a lot of reference builds are possible. So for example, AG38 or yeah, AG38 is kind of the second latest version. The latest will be T2T. So yeah, this is if you go to NCBI, you can find this genome. This will be in FASTA format. So I'm not going to go into detail of FASTA format. You can just look it up. It's a very simple format. There also you'll have the sequence and the position of the sequence. So generally, if you look at the length, it's around 3 billion, 3.1 GB. Ungap length is 2.9 GB, so 0.2 GB is still gap. And then you have number of chromosomes. 24 would be including the Y chromosome as well. And basically, what is the N50 length, what is the assembly level, and so on and so forth. So extra information: how many total genes are there, how many protein coding genes are there, and across each chromosome, what is the length. So, for example, uh, in this is chromosome 1, the length is around 248 million base pairs, and the GC content is this 41.5%. So across all the chromosomes, you can get this information. And also there will be mitochondrial DNA involved as well. So all this information you can get in a FASTA file. And just if you go to the FTP site, this is the file .fna.gz. If you download this, you can just see. Uh, so just let me go to the README here. .fna. Yeah, yeah. This will be the FASTA format of the genomic sequence in assembly. There will be masking. The genomic fna.gz file will contain all top level sequences, including the assembly, the chromosomes, plasmids, whatever. And basically, it will have the entire genomic sequence. Now, coming back to what we were discussing. So you have, this is your output, right? The read that has been generated. Now you have to find where this read has come from the entire genome, because you do not have that information from where this read has originated. So what we do is something known as alignment. This process is known as alignment, finding where this stretch of sequence is there in the entire 3 billion base pairs. So as I was saying, so Illumina generates how many, like 500 base pairs length of DNA, and your entire DNA length in the human genome is 3 billion. So what you're doing is, from a computational perspective, you're doing a string matching problem, right? You have a string of 500 base pair, you have a string of 3 billion base pair. You have to find the location of this 500 base pair in the 3 billion base pair. So if you do it using traditional techniques, using traditional algorithms, this problem will not like you cannot do it. Like technically, the amount of computation power required and the time that will be required is not feasible to do. So you need very sophisticated algorithms to handle this kind of things. So we are going to talk a little bit about those things. Now, just quickly, I want to focus on why you're doing alignment is to also definitely to find where that read has originated from, but also to see if there is any mismatch with the reference genome. So, for example, you have a 500 base pair long read, 499 bases are matching, but one base is a mismatch. That will tell you if there is a mutation or not. So, for example, let us take this particular example. So, the colored bases is your reference genome. These reads have been generated from your sequencing experiment, and you see here at this particular location there are how many reads? One, two, three, four, five, six reads supporting this particular location, right? So in the reference genome, there is a T, but two out of the six reads that you have generated are telling you there is a C there. Right? This might be a mutation, right? We are not definitely sure from here. We have to check a lot of contexts, right? So if is the read properly aligned? What is the level of evidence that we are seeing? So what is the quality of these two reads? So there can be different levels of quality. The quality of the base pairs here, as I was saying uh, earlier. So these are the quality of the base pairs, right? A, F, F, F. These are the quality. Say, for example, there is a mismatch at this particular position. Say, for example, this particular T and the quality, and just this is ASCII code, I'm just assuming. Say, for example, this quality of this base pair is very low. Then even if you have a mismatch here, you will consider that to be more of a sequencing artifact than an actual mutation based on the quality parameters. So there will be a lot of context involved, but if generally if there is a mismatch like this, you will call this a particular variant. This will be a mutation. This will be a variant site in the genome. So what type of variants are there? There could be single changes. So there from a T there is a C. Uh, so for example, this is person one and person two and person three. So these are their maternal and paternal genomes. So in your data, so for example, you have sequenced person one. You have seen since DNA is double stranded, you are seeing half the reads supporting a C at the position, half the reads supporting a T at that position. So there has been a mutation there. So single base pair changes, those are known as single nucleotide variants or SNPs, SNPs in general. And we determine the frequency. Not all these SNPs will be disease causing or harmful in nature. Some of these are just benign. They do not cause any problems in gene expression or protein production or cause any diseases. This will be just benign. So how to determine if the variant is benign or pathogenic? We have different techniques for that. But generally, the idea would be that if this variant is very prevalent in the population, so if you sequence, say, for example, 1,000 genomes of 1,000 human individuals, and then you find this variant is present in, say, for example, 500 of them, and all of them are healthy individuals. So basically, this variant is not contributing to any disease or anything. This is a benign variant, right? So the effect of that variant will be determined based on how frequent it is in the population. If it is very rare in the population, if it is less than, say, for example, 0.1% of individuals are carrying this variant, and each of those individuals carrying this variant are also showing some sort of phenotype which is corresponding to a disease, then it becomes important. Other than that, it is a benign variant. But generally, this is how you determine if there is a variant present in that location or not. Now, that is a single base pair change. What can also happen is some portion of the genome has been deleted, some portion has been inserted. There are repeat units. These kind of uh, uh, variations can also occur. I'm not going to go into germline and somatic mutations. I hope you guys know that about a little bit. Now let's go back to the actual alignment problem. The alignment is one of the major issues there, right? So you have a smaller base read of length 500 base pair, you are trying to align it to a read of 3 billion. That is a problem. So that is what the alignment problem defines. I'm going to go a little bit mathematical here. You do not need to completely understand all the equations and everything. It's just you should get the idea about generally what is happening. Right? So yeah, the alignment problem is similar for the DNA alignment, or also if you do protein alignment, amino acid alignment of two proteins together, that similar kind of a problem. Right. So the input would be two sequences like this, X and Y. You need to find the optimal alignment between these two sequences, or basically form a column of type. So basically, C and so these T are matching A and G, there is a mismatch, and so on and so forth. So basically, two sequences are not of equal length, right? So you can introduce gaps, you can introduce extra characters, and then you have to find what is the best possible match, right? So how do you do that? Uh, uh, like before coming to how we do that, when there is a mismatch, right? When there is a mismatch, it can mean mutations. As I was talking about, it can be a substitution. A single base pair has been substituted. C has been substituted with T, or a certain number of base pairs have been deleted, a certain number of base pairs have been inverted, and so on and so forth. A lot of things can happen. Uh, so what happens if a single nucleotide deletion or insertion occurs in a coding portion of the genome? What happens if a single nucleotide substitution occurs in the coding portion of the genome? So this uh, will be a substitution where Xi and Yj are matching at that position, but at that position, the nucleotide, there is a mismatch. In this case, there is a position in X, but there is not a position in Y for the same location. So there is a deletion in Y, and opposite, there is no position in X, but there is a base in Y. So that means there is an insertion, right? So there we use some sort of scoring techniques to tell uh, if there should be a gap or there should be a mismatch. So generally, this is what is known as a scoring matrix. This is the very simplistic form of it. So the idea is that if you have a match, you use a +1. If you have a mismatch, then you use a penalty of -1. And if you introduce a gap while doing the alignment, you use a -2. So this is the very basic yeah, scoring technique that you can use. Sorry, scoring matrix. Now, for example, these are two strings that you are matching. So there is AAAC and there is AGC. What will be the best match? You can do multiple combinations. So one will be introducing a gap here. The next would be the introducing the gap at the end of it. So if you do, so first there is a match scored A comma A, then there is a gap, then there is a mismatch A comma G, and then there is again a match. And if you add that up according to this scoring system, you get a -1. And if you do the second technique, which is like introducing a gap at the end, if you add the scores up, you get a -3. So -1 will be greater than -3. So this will be a better alignment than this, right? So according to the scoring scheme, you can tell if the alignment is a good alignment or a bad alignment and which is the best alignment, because there will be n number of possibilities. Computationally, it is not possible to like kind of bring out everything. So what kind of algorithms do you need? So if you just try brute force, if you try all the combinations, right, all possible alignments, score all alignments, and then select the best alignment with the maximal score, that will take exponential time, and in real sense, it will not finish the alignment, right? But we need to do the alignment, like this cannot be the bottleneck here, right? So one uh, software that uses kind of a brute force technique is BLAST. It can, but it does use a brute force technique, but it uses additional data structures which help it save time and memory. So it uses a lookup table. I'm going to come to that a little bit later. What a lookup table is, what is indexing, and so on and so forth. But it uses lookup tables, which are additional data structures which help in searching for these um, for these reads in different locations. Yeah, I'm not going to go into BLAST. You can directly look at that. You might all, like, if you're coming from a bioinformatics background, you must have studied BLAST. If not, like, if you think like I should discuss BLAST, I can do that in the next classes. Let me know about that. But right now, I'm skipping it. Okay, now the idea is, if we have very distinct regions of the genome which have distinct sequences. What we can do is we can index these sequences. So we know that this sequence has come from this location if you have the original reference genome. So what we do is we index the original reference genome. So generally how indexing works is like how indexing works in a book. Uh, it it's basically most books will have an index where a word will be mapped back to a page it is found, right? Here also similarly, you have a sequence which can be mapped back to the location from which it has been generated. So generally, this is how the indexing will look in a book. So, for example, the word "the" is present in these many locations. The word "cow" is present in these many locations, and so on and so forth. So, this is a straight index, forward index, right? And if you invert the index, so basically you do it the opposite. In document one, what are the words, right? In document two, what are the words, and so on and so forth. So, this inverted index is used for generating the index of the reference. So before doing the alignment at all, before doing that alignment at all, you index the reference, to know that which locations have which kind of reads. So that helps the alignment drastically, right? Okay, so that is indexing. So generally, how the indexing occurs is you create substrings of a larger string. So if this is a larger string, you create substrings of this. So this will be the first substring. Second will be one overlap, then third will be again one overlap. This is a substring, and then you put offsets. So this has come from offset 0. This is coming from offset 4. This is probably coming from 2 offsets, and so on and so forth. So offsets are telling you the location from which uh, uh, from where the read is getting generated of length what. So basically here you're only indexing reads of length 4, and then you sort it and tell that this index has come from this location, then this index has come from that location, and so on and so forth. And just to make it clear, these are computationally very challenging things to do. I'm just telling you in a very easy manner, but these are very challenging things to do in a with respect to a data structure. So basically, you use hash tables and other kind of specialized data structures to generate these indexes and store them. So uh, anybody of you who has taken up data structure and algorithms are aware of time complexities and space complexities. Here, both are an issue, right? Because we have 3 billion bases, so the space required to store that data and store the index is also very large. And also to do the actual alignment, the compute needed to do the actual alignment is also very hard. So both the space and the time need to be optimized to the maximum to get the output, right? Yeah. Okay. So yeah, generally you see the indexing. Sorry. Sorry. This- Yeah, generally you see the indexing times are very high. So for example, an index of the human chromosome of around chromosome 1, so which is around 249 million bases, will occupy 12 GB of space. That is only for one chromosome, right? So it's not very ideal. So you need to optimize that as well. So you generally do that. Okay, now you have the indexes. Now you have to find the actual alignment of the indices, right? So uh, so you have these uh, you find these indices, and then you see so you look for a particular pattern in these indices, and some of these will be fruitless and they will not correspond to any matches of them. So P is a pattern, T is a text. So in the text, you're trying to find the pattern. So the text is your reference, the pattern are your reads. So some indices will not have, but some indices will have. So there will be a list of indices corresponding to your read, and then you can go map and go back to those locations and check for the reads itself. So yeah, I'm not going to go into these details. Let's just skip it. Huh. One data structure I want to talk about is the trie data structure, which is very commonly used. Trie, trees, and suffix arrays. Right. So generally, what we do is, uh, uh, so for example, yeah. So for example, you have a text like this. This is a very long text. So this is your text, and if you will be told to find a pattern within this text to know the location from which a pattern from this text has originated. So how we generally go about it with respect to building suffix tries and suffix arrays. So what we do is we add a symbol to the end of the look end of the read sorry end of the text, which will tell you that this is the termination of the text, and then we generate suffixes of the text, right? So the first suffix will be just if you remove the first element, that will be the first suffix. So this is the first suffix. Then you remove the second element, this will be the second suffix, and so on and so forth. You go on until you reach the termination. Uh-oh, sorry for this. Yeah, you go until you reach the termination signal, right? So these are all the suffixes. All of these are suffixes to this single text. Now, taking these suffixes, what we build is a suffix trie. So from the root to the node, you can directly you can get all the suffixes of this particular text, right? So this is the text, all the suffixes are there. You build all the suffixes into the suffix trie. The idea is that if you start from the root and you travel to the leaf of the tree, you will get all suffixes. So one suffix of it would be BA dollar. So that would be this one. Okay, I don't think these are the same, but yeah, so this is a separate example. We are taking this text is this. So uh, if you yeah, so if you start, say for example, you want to find so if you start from here and you follow this part, so from the root, all the edges will be labeled as the elements. So you start from here, from the root, you travel to one of the leaves. So this is one of the leaf. So you would start from the root, BA dollar. So that is one suffix, which will be this one. The next would be this whole path, BAABA dollar. So this would be this one, BAABA dollar. Like that, you can incorporate all the suffixes of a particular string into a data structure like this, which is known as a suffix trie. Now you see there are a lot of redundant edges. I can just combine all these three edges into one, right? So here is one edge for B, one edge for A, one edge for dollar. I can just combine it into a single one where the branching is happening. So here the branching is happening. Here uh, the the suffix will be different, but till the point the branching is happening, I can just collapse it into one. So I can just have one node here, and it in the edge I can have A B A dollar. Right? So that will crunch up the space even. Right? So this is known as a suffix trie. If you crunch it up, this is how it will look. Right? You crunch all the non-branching labels and you into one node. This is how it will look. So this is known as a suffix tree. So looking now, when you look for substrings in a suffix tree, what you're going to do, you're going to start from here. You're going to match one element. So for example, you want to find BAABA in this particular, right? So this was the whole string, right? In this, you want to find BAABA, which is like all the elements except A. How are you going to find it in a tree, suffix tree? You start from the root, you then match one character after another until and unless you reach a branch. So start from here. First BA is matching. Here you have to make a decision. Do you go here or do you go here? So you do an alignment on both the sides. In this case, it will be BA dollar, which is not matching your string. But on the other side, if you add this, so BA, ABA dollar. So this will be a complete substring of the uh, original uh, reference, right? Original text. So that is how you can get substrings from a text. Sorry, if you have a very big text and you want to find a small pattern in there, generally this is the technique that is used. You build a suffix trie, collapse it into a suffix tree, and then you do a path traversal. So this is a walk in terms of graph theory. You can call this a particular type of walk. You start from the root and you end in the leaves. So it'll tell you if there is a suffix that actually corresponds to the original text. Yeah, so yeah, I'm going to skip all these. These are like a little bit more computationally developed things. So how do you find the longest common substring? All these are like alignment problems. Longest common substring, right? It's the same thing. You have a read. That read is actually a substring of the original reference. How are you going to find where is this the longest common substring? Right. So that is also a problem. Those things I'm not going to get into all the details because yeah, it's a lot more uh, like complicated. I'm not going to do that. Uh, yeah. I'm going to just quickly touch on suffix arrays. It's very similar to suffix trees. It is even a better space optimized data structure than sub trees. So if you have arrays, you do not even have to store the suffixes. You just store the offset of the suffixes, location of the suffixes, right? And you store the suffixes in a separate data structure. So this becomes very, very uh, com computationally nice, like nice to work with. So, but even suffix arrays uh, are not the best. It will occupy about 12 GB uh, from 47 GB. If you use a suffix tree, the index will be 47 GB. If you use a suffix array, it will be 12 GB, but it's still not the best one. But yeah, we are moving one step closer to the best algorithms, right? This I'll skip, but this is actually very important. Burrows-Wheeler transform is the technique that is used to create these indices and compress these files. So I'm not going to go into details, but yeah, you can compress a file by creating the Burrows-Wheeler transform, and then you can just get it back. So this logic is also used in the alignment problem. Yeah, that's all. Mostly, I'm not going to discuss the Burrows-Wheeler transform, but I'm just going to give you the paper. I think if you go through that, think it can. Uh, yeah. So this is an algorithm that uses Burrows-Wheeler transform to do the alignment. So the alignment algorithm is known as BWA, Burrows-Wheeler alignment tool, and it uses this Burrows-Wheeler transform to actually do the alignment. So this actually talks about the algorithm, they bring, how they build the suffix trees, how they do the transformation, and how they actually get the alignment from the transformation. This actually is Actually, and this is an older paper. You can see this is a 2009 paper. This used solid sequencing, by the way. This did not use, yeah, this did not use Illumina or anything. It used data from solid sequencing to generate this algorithm. So interesting. You can give a read if you are interested. Okay. Any questions before I move into the assignment questions? I have talked a lot today about a lot of things. A lot of things you might be getting confused with. Do not get confused. I just wanted to give you an idea about how the things actually work in real life with real life data, and to give you an idea about what the level of sophistication in terms of computational algorithms you need to work with this level of high throughput data. The generation of high throughput data is very good. We have come a long way here. We The cost has gone down a lot. The throughput has increased a lot. Uh, Along with that, we have also developed a lot of very efficient algorithms to actually handle the data. Otherwise, like if you want to do, as I was saying, like the first step would be alignment, then the next step would be variant calling, right? So, and in between, there are a lot of steps of quality control. There are a lot of other steps as well. So generally an end-to-end pipeline, if you start with the raw data to get the variant calls and what genes these calls lie in, if they're in pathogenic variants or not. Uh, this takes a lot of time. For a short read data with the best of algorithms, it'll take you 45 to 50 hours for one sample. So it's more than 2 days at a like running at a HPC, like at a supercomputer level. Not even like these things cannot be run on, say for example, my system. This is a MacBook. This cannot run all those algorithms efficiently. You need very high memory. You need very efficient s- uh, like algorithms to run it. So yeah, I just wanted you to make you appreciate the fact that a lot of effort has gone in to actually develop these algorithms, and this has led to where we are standing today with a lot of data, a lot of analysis, and so on. Okay. Yeah, reference-based sequencing, I'm not sure, but like the alignment is toward, so the idea is that what you're generating in terms of the data from the sequencing experiments are these short stretches of DNA sequences, right? 500 to 700 base pair if you're doing Illumina or other techniques, all the short read techniques. 500 to 700 base pairs of these short stretches of DNA. And now what you're trying to do is finding where these short stretches of DNA have come from in the original genome. So the original genome will be the reference genome, and you're doing the alignment of these reads to the reference genome. So yeah, I'm not sure what you mean by reference-based sequencing. All the sequencing will be so okay. If you are talking about assembly-based, this is not assembly-based. Yeah, if you mean it like that. So we are not assembling the genome. We have a known reference genome. We are just aligning our reads to the known reference genome. In the Human Genome Project, there was no reference genome. They did assembly, right? What they did was like also using very sophisticated algorithms. They generated reads which were overlapping with each other, and using these overlaps, they generated the entire sequence, right? For for there you use like different graph theory techniques. So you build these overlapping graphs, and then you do some sort of Eulerian walk or different kind of walk to go from one point to another. And if you can travel the whole graph, you'll get the whole sequence. So that is assembly based sequencing. That will be like you do not have a reference genome. You are generating the genome directly from the data itself, which will be a lot of error will be there if you do it just based on short read, low depth coverage data. But yeah, generally what we do is we generate the data, but we align it to a reference. We do not do assembly. Any more questions? Okay, then if there are none, we'll move on to the assignment questions. Okay. I'm not sure what is, sir, please leave. Acha, please leave reference to learn this content. Uh, I'm not sure if they're like direct references. I follow particularly a few people. Uh, One is known as Ben Langmuir. He's a very well-known professor at Johns Hopkins University. He's a professor of computer science. You can check out his lectures on YouTube. He teaches very well, and there is also a website also. He's the actual one of the authors of BWT. So he's one of the original people who actually developed these algorithms. You can check out his website. Uh, Other than that, I also follow this another person, Manolis Kellis. He is he's a professor at MIT. He teaches a lot of population genetics and a lot of other computational genomics courses also. You can check out his content as well. These two are majorly in terms of teaching. These are the people I follow, and they are kind of the best in the field. And if you find this a little bit like a little at a higher level, you are not being able to understand or anything, I'll suggest there are other NPTEL courses as well, where we just talk about NGS data analysis. I think there is a course from IIT Kharagpur offered by Professor Ridhiman Dhar. I was a TA in that course for one session. So that course just deals with data analysis, how to do NGS data analysis across different modules. You can take a look at that course as well. And other than that, I think these two people, if you want to learn about algorithms used for these alignments and other technology techniques. Ben Langmuir is the person. If you want to get a general idea about how sequencing data is used, how it is, how it is interpreted, how it is analyzed, Manuel Scanlis has a lot of courses on YouTube. Both of them have a lot of courses on YouTube. You can just check it out. A lot of my material is also based on their courses. So yeah. And is there any existing I'm not aware of any existing Telegram groups or WhatsApp groups. If you want to know about this kind of stuff, you have to ask it in the discussion forum. There are other TAs which handle those parts of the course. They will able to help you. I just take the 2 hour live sessions. I do not have any more engagement with the NPTEL course. So if you have any doubts outside of these things, like the things that we discuss, you can directly post a question on the discussion group. I'm not sure if there is any Telegram group or not. Yeah, this is my email address. Okay, any more questions? Okay, then move on to the assignment questions. I think we are running a little bit late, so I'm just going to uh, hurry up a little bit. Okay, uh, I'll just be back in a minute. I just need a water break. Uh, I'm audible, right? Just a thumbs up or a yes. Yeah, okay, thanks. Okay, yeah, somebody I think said option B, which is true. You do not do denaturation of DNA in actually any of the sequencing technologies. You mainly do denaturation. You have a double-stranded DNA, you break it down into single-stranded. Generally, do not do denaturation. So, yeah, that's the correct option. Moving on. Uh-oh. I think some of it is marked. Okay. Anyway, the answer is already marked here. So, So the question is, how many hydroxyl groups does a single isolated DDNTP contain? So these are the structures of a DNTP or a DDNTP. So generally, this is the R group. In DNTP, you'll have a hydroxyl group here. In a DDNTP, this hydroxyl group will be replaced by a hydrogen group. So generally, you have 4, 5 in DNTP and 4 in DDNTP. So 4 should be the answer. Okay. I hope that's okay. No? So an extra OH in DNTP with where there will be 5 hydroxyls. If you remove this OH with a H, there will be a hydrogen here. Yeah, yeah, sure.
Excuse me, please. Yeah, please repeat the question number two. Just like I've missed it.
Oh, the question is how many hydroxyl groups does a single isolated DDNTP contain?
Okay, thank
Yeah,
you.
I skipped this because by mistake I have put the answer here here itself. So yeah. Okay. Yeah, that's correct. Emulsion PCR. So basically, you have the it connected to the beads, the streptavidin beads with the biotin labeled DNA sequences. So yeah, you do emulsion PCR on that. Okay. Okay, I'll wait another 30 seconds. Okay. Uh, the answer is basically all of them. This, I just wanted to remember that Sanger sequencing is still the gold standard in terms of accuracy. So what happens is if we say, for example, we are doing long read sequencing, even if we are doing nanopore, we have found a particular variant, and we know that that variant might be causing a disease in a particular patient sample. But to confirm the presence of the variant, we still go back and do Sanger as a validation along with long read data, right? So whatever be the throughput, whatever be the sequencing technology, the accuracy cannot be beaten of Sanger. Even today, if you want to validate a particular variant, you do Sanger. But again, you cannot do it for like millions and millions of variants. But if you have a candidate 1 or 2 or 5 variants, you definitely should do Sanger and validate it. So all of them are actually having lower accuracies than Sanger. They are having a lot higher throughput, but accuracy wise, all are lower than Sanger. So just remember that Sanger is still the gold standard. Which one would have been the highest error rate? I would guess nanopore. Illumina, so basically the error rate also scales with the length of the read. The more the read length, the higher error rate you are going to have. So the longer reads, PacBio and Oxford Nanopore have higher error rates, at least at smaller nucleotide levels compared to Illumina. So yeah, so basically throughput and read length, both of them actually with increase of both of them, the error rate increases. So I would say a nanopore will have the highest error rate. Okay. So in this one, uh, yeah, what are the advantages of 454 sequencing compared to Sanger or chain terminating? So obviously higher throughput than Sanger. No labeling of nucleotides is required because in 454 you are using pyrosequencing. There is the enzyme luciferase which will convert it into luciferin and give up the light. No labeling required. Also, no need to perform gel electrophoresis, which is often the case, which you have to do for Sanger. So, and uh, yeah, the error rate will be higher of 454 compared to Sanger, right? So that is definitely not an advantage, but yeah. Okay. Okay. Yeah, that's correct. It uses sequencing by synthesis with reversible termination. So yeah, that's correct. Moving on. Okay, so AC and an AD. Okay, we'll wait for another 30 seconds. Okay, okay. Yeah, the answer is higher read length. Right. So as I was saying, 454 kind of sits in between Illumina and the best Sanger would be around 700 and long read will be above 1 KB. So 454 kind of sits in between that. So yeah, it's high read length. Throughput is higher for Illumina. Illumina is faster and basically 454 has more homopolymer issues than Illumina. Illumina actually is one of the best for homopolymer regions. Yeah, but 454 is not, so definitely. Okay, moving on. Yeah, this question, before you answer, I think I should discuss the answer of this because this is, a tricky question in terms of this question is fully not correct. So uh, in in the assignment page, the answer given is I think 454. Uh, but the question is irreversible chain termination for DNA sequencing actually is not used in any of these four. But the answer given is 454 in the assignment page. But I just want you to know that so Illumina and first Illumina and Solexa, they are the same thing. Basically, Solexa technology was bought by Illumina, and it's basically the same thing. And they use cyclic reversible transcription. So definitely not these two. Helicos also use something called virtual terminators, nucleotides that in fact retain a 3' OH but carry a bulky dye linker that sterically hinders a second incorporation until it's cleaved. So it is functionally reversible and it is not irreversible. 454 does not use terminators of any kind. It allows natural incorporation of DNTPs at uh, one uh, one time, one at a time, and you can detect it with the respect of the fluorescence emitted by the enzyme. So this is also like not using irreversible chain termination for anything. So across all the four um, options, the option given in the assignment is 454, but I do not completely agree with that option. I don't think any of these four are using irreversible chain termination. What you can say is if you add Sanger here, then it makes sense. Sanger uses irreversible chain termination. But other than that, all of them are using sequencing by synthesis and reversible termination. There is no termination per se, right? So, yeah, this question is a little bit tricky. I just wanted to say that I think none of these options are correct, but in the assignment page, the second option is given as correct. So, yeah, okay. Yeah, I'll move on to the next one. Yeah, the answer is correct. Proton hopping is basically a hydrogen ion will like kind of move through a network of water molecules as a chain reaction. So yeah, hydrogen can do this proton hopping. So and basically the ion current sequencing will detect the pH change due to this. So yeah. Yeah, it's all to do with the throughput and the speed. It's nothing in terms of homopolymers or long reads or anything. It cannot generate that. It's just that yeah, it is faster and with higher throughput. That's all. Yeah, correct. So moving on. Yeah, you definitely need to revise. Yeah, I see one answer. Yeah, it's correct. All of them use sequencing by synthesis. All of them use DNA polymerase. So that's correct. Okay, I have one answer. Wait for 30 seconds more. Okay, the answer is, I think, D. Okay, so you have to remember this is a double-stranded DNA, right? So you do not sequence double-stranded DNA. You sequence single-stranded. It has mentioned it is double-stranded. So basically, single-stranded would be 4250, and there, yeah. So you will have magnetic adapter to be of 244 Daltons. You'll have three of them, one to each side and one to attaching the probe itself. So 3 of for 244, that will be 732. And if you add this to the single-stranded DNA mass, it is 4982. The trick thing here is the double-stranded DNA and 8500 antennas. You never do sequencing with double-stranded DNA. It's always single-stranded. So these are the tricks you need to keep in mind. In these kind of questions can come in the examinations as well. So yeah, keep this in mind. It's not double-stranded. You do it single-stranded. And yeah, yeah. Okay, moving on. Yeah, the answer is sorry. Mm, yeah, not A, by the way. Right. I think somebody mentioned A here. In pyrosequencing, you do not label the nucleotide with fluorescence. Due to the incorporation of the nucleotide, some chain reactions are like coupled reactions are reactions go on which generate the light which you capture. You do not use labeling, fluorescent labeling for pyrosequencing. On the other hand, for Illumina, you have to label the nucleotides with fluorescence. And also for helicose, because they rely on the optical image of these fluorescent dyes. So pyrosequencing is natural light from cruciferous. It's not labeled dye. So I think somebody answered pyrosequencing, which is not correct. So keep that in mind. Okay, moving on. Yeah, helicose is something that we have not covered. Probably in the next session, I can just give a quick overview. I think that would make sense. I'll write it down. Next week, I'll give a quick overview on helicose. Yeah, I think somebody answered all right. Yeah, that is correct. Yeah. So the beam stands for beads, emulsion, amplification, and magnetic. So yeah, all of these are involved. So, okay, this should be quite easy. Yeah, I'll not wait for the answers and like yeah, option C is correct. You add the DNTP there, the gene termination occurs. No more DNTP can be added. So that's correct. Yeah, complete genomics is also something that we have not covered. So yeah, I am also not fully so I am also not fully aware of what complete genomics platform is. So I have not covered it in my session, but I think Sir has covered a little bit of it in the in the lectures. So yeah, yeah. The answer is that palindromic sequences and generating single molecule reads are basically the challenges for complete genomic sequencing. So complete genomics is basically another, just like 454, this is another sequencing technology, but it's very similar. It also does sequencing by synthesis, but I'm not fully aware of all the details. So that is why I'm not going to go into very details of this. But yeah, these two are the answers. Yeah, so it definitely cannot produce long reads. This is not a long read technology. Uh, but yeah, it struggles with complex palindromic sequences. Hope you know what palindromic sequences are. So it's the sequence which reads the same from 5 prime to 3 prime, from 3 prime to 5 prime. It's the same sequence. So yeah, yeah, it is also faster. I think B and C, somebody mentioned. Yeah, A is also involved. It is also faster, basically. It just cannot produce the longer stretches of DNA. That is only done by PacBio, Nanopore, and other long read technologies. All of these are short read. So yeah, it is faster, it is higher throughput. And homopolymer, it is better than pyrosequencing, but I don't think it is as good as Illumina, but it is better, certainly better than pyrosequencing. Yeah. Okay, moving on. Yeah, again, a complete genomics question. I'm not going to go into detail of this, but yeah, it uses rolling circle amplification and produces a DNA nanoball, basically. That is the USP of it. So it creates a long repetitive strand of DNA that folds into a dense DNA nanoball. So yeah, rolling circle amplification is the answer. Okay, moving on. Yeah, this is also related to complete genomics. What is the diameter of the DNA nanoball? I'm not going to like I am not fully aware of this technology, so I'm not going to give my opinions on this. Just what is the question? What is the correct answer? Yeah, sir has discussed this well, so yeah, nobody is answering. So the answer is 300 nanometer. So yeah, the diameter of the DNA nanoball would be around 300 nanometer. Okay, moving on. It's the final question. Okay. The answer is the first three. Sorry, first A, B, and D, not the five. So in solid sequencing, the probe is an octamer. It has a three prime end that gets ligated to the growing change, two query bases that dictate which fluorescent dye is attached to the five prime end, and an internal chemical cleavage site. So the cleavage site will be there, the fluorescent dye will be there, and there will be a three prime ligation site, which will be at the five. Yeah, because the three prime end will be ligated, right? So it will move from five prime to three prime, but three prime end will get ligated so that there will be a three prime ligation site, there will be a cleavage site, and there will be a fluorescent dye. So that's it. Yeah. So that's all from my side for today's session. If you have any question, anything to discuss, we can do that now. Okay. If there are not any more questions, we'll close the session. Somebody, I think, was asking about the slides. The slides will be available in the folder. It will be shared with you from the NPTEL team. The videos are uploaded on YouTube. That is also that folder where all the links are present. That will also be shared. Yeah, that's it. Okay, thank you guys for joining, and we'll see you next week, Tuesday. Thank, thank you.